Good evening, everybody. Thank you very much for asking me to talk. 15 minutes neuroimaging and schizophrenia. Gulp. I'll do the best that I can. I just want to tell you a little bit about my background first, though. I'm a clinical scientist and physicist by training. I currently work in the nuclear medicine department at UCH. Previous to that, I was working in psychiatry at the University of Cambridge, which is where most of the stuff that I'll talk about now comes from. So, as a scientist, I understand that it's an understatement to say that schizophrenia is an incompletely understood disease. However, contribution from neuroimaging has shown that the disorder involves abnormalities of some specific neurotransmitters, such as dopamine and glutamate, and that there are definitely structural and functional abnormalities associated with this syndrome or collection of symptoms. And I think the other thing that we've shown is that the neuroimaging abnormalities in schizophrenia are present to a varying degree, and I think this is a reflection of the different types of symptoms and the variance in their severity of their disease. And that you have to understand that currently neuroimaging techniques are not diagnostic of this disorder. So what I'll be presenting next is group average. So I say way back. A few decades, the first thing that people started to notice that there were very profound structural differences between people who were said to be diagnosed with schizophrenia and normal. And that was in the size of the ventricles, as shown by this very old basic MR scan, but also evident in X-ray CT, I believe the first reports were done. And then we started doing things a little bit more sophisticated. So we were able to take high-resolution grey matter images, T1-weighted high-resolution images of the brain, and start doing quite sophisticated post-processing and be able to produce these voxel-wise maps of grey matter differences between healthy normal controls and people who are said to be diagnosed with schizophrenia. And these areas in red are showing statistically significant losses of grey matter volume. So I think you would probably understand this as being an enlargement of the sulci, an atrophic looking brain in comparison to age-matched controls. And then we moved on to even more sophisticated mathematical modelling of this, where you can take these grey matter maps, segment them into anatomically labelled regions of the brain, and produce these covariance maps where you're correlating voxels against one another across the entire brain, and then threshold at some sort of R value. I hope you're familiar with some of these statistical terms I'm using. And then start to pull out graph topography measures, such as the number of nodes, the density of connections to each node, defining hubs. And these coloured blobs here are what would be called the hubs of a network. And to show that these are normal controls compared to people, a group of people who are said to have schizophrenia, there are quite marked differences between the topography of the network. And this is where, I'm talking about group average differences rather than being able to do this in a single person brain and then say, yes, they've definitely got schizophrenia because it maps onto this network. We're not there yet, but we're trying. Okay, so I've talked a little bit about structural imaging. Functional imaging, quite a few of you, I imagine in psychology, are quite familiar with this functional MRI, where you're measuring the neural activity of someone's brain while they're lying in the scanner. We use this using a time series MR signal that is very sensitive to the oxygen concentration in the brain. And you can do things like just measure their brain activity while they're lying in the scanner. And I've used an EEG signal to kind of be a proxy for that, or some sort of neural stimulus where you show them a section of images and then nothing and then a section of images, and you can mathematically model the changes in signal with this step function. And using this sort of idea, people have been able to show that there are differences in the way people's brains function when you compare them to healthy controls compared to a group of people who've been diagnosed with schizophrenia. This was people who, I've got the normal around, sorry, that's the schizophrenia, this is normal controls. Auditory presented emotional stimuli. So here you can see that there's a hyperactivation of the brain networks involved in this cognitive task compared to healthy controls. I'm not a clinician, I can't interpret this result for you, but I'm just flagging this as a way that we can study how people's brain function also is affected during these pathological conditions. Slightly more sophisticated, along the lines of the graph network topology that I talked about, now you can do covariance maps across time. So here we've mathematically deconfolded these signals here so that we can start looking at particular bandwidths of activity and correlate them voxel-wise across the brain. Then we can map them back onto a brain space, and we can do a colour-coded map of the strength of correlation or covariance between activity as measured by oxygen concentration in the brain of healthy controls and schizophrenia. Here, this basically says that there are stronger connections between certain areas of the brain and healthy controls compared to this dampening down of connections in schizophrenia. Again, I can't really interpret this. This is a fantastic paper by one of our medical students that she did a summer project with us and produced this paper. I do recommend you read it for an understanding of graph networks and time series and resting state activity. Okay, so now we leap on to yet another form of MRI imaging, diffusion tractography. I'm not sure if any of you are familiar with it, but you can sensitise the water molecules and the direction in which they're travelling in the brain so that you're mapping the anisotropic movement of water molecules along nerve fibres. So here, this is the kind of map that you'll get out from the MR scanner, and the red is going up and down, the purple is going left to right, and green, I believe, goes up and down the brain. You can then use, again, some more sophisticated mathematics to start drawing these tracts. So that's where the tractography term comes from. You can see all these white matter bundles and the direction they're going into in the cortex. And there are hundreds of papers out there now, but this is one of the early ones about showing the differences in the what we call white matter integrity in schizophrenic subjects compared to normal controls. And there's a lack of integrity or degradation of this anisotropic movement of water molecules within schizophrenia compared to normal controls. And finally, one other MR technique, and this is where we get into the neurotransmitter side of things. This is magnetic resonance spectroscopy. So this isn't so much about looking at images, but measuring the chemical shift in the hydrogen atoms that are in different molecules. So if you cast your mind back to your chemistry A level, there'll be a chapter in there towards the end of the book that talks about that you can measure magnetic resonance frequencies of the hydrogen protons will be shifted slightly depending on what their surroundings are. And in this case, so you produce a spectrum. So these are looking at hydrogen molecules, hydrogen protons in the brain, and we can plot them out for different molecules. This one is for the NAAA and acetyl aspartate, which is linked to glutamate receptors, and showing that they're reduced in this particular area of the brain, because that's where we looked, in schizophrenia compared to normal controls. This is still quite a nascent area, quite exciting. Okay, so I've talked about magnetic resonance imaging. Now I'm going to talk about something else, positron emission tomography or single photon emission tomography, where you inject your subject with a tiny, tiny amount of radioactive tracer that's labelled with usually fluorine-18, sometimes carbon-11, and you attach that to a particular pharmaceutical that will go to, will either be metabolized or go to a particular neuroreceptor in the brain. And then we put the people in the scanner, and these crystals will detect where the gamma rays are emitted from the brain, and then we can map these into brain images. If you want me to go into this in more detail, we can talk later on. Okay, so this is one example of looking at nicotine receptors. So they asked a group of healthy controls who smoked, and schizophrenia, subjects with schizophrenia who also smoked, which apparently is like 99%, I think, to not smoke for about 24 hours, and then they injected them with a radioactive tracer that was stuck to a pharmaceutical that was avid for nicotine receptors. and showed that there was a lack of nicotine receptors available in the schizophrenia subject compared to normal controls. Make of that what you will, but I understand if you're thinking about how your antipsychotic drugs interact with the brain, you start getting some clues when you look at the neuroreceptor density that's available. I like this slide because it makes me understand how the drugs work. This is a healthy volunteer who's been injected with a tracer that's avid for dopamine receptor areas in the brain. In the basal ganglia, there are a lot of dopamine receptors. It's the part that goes wrong in Parkinson's disease. If you give this person risperidone and then another injection of radioactive tracer, there is nowhere for the tracer to go to be uptake. You completely blocked your dopamine receptors at that point with risperidone. Olanzapine, less so. Clozapine, less so. And as I understand that if there is severe extrapyramidal side effects, you would swap from risperidone for clozapine. I might be way out of date on these drugs, so please fill me in later. I'll be interested to hear what the latest version of antipsychotics are. If you can't tell the difference between whether or not it is a part of side effects of the drug that your patient is taking, or it is the beginnings of Parkinson's, you can use some dopamine transporter imaging. So that's looking at the other end of the other side of the cleft. And you'll see that if it's drug-induced, their transporter system is absolutely fine. So you've got this is called fluorine FBCIT, but it's avid for transporter system. In patient with Parkinson's disease, there's no transporters left. It's all gone. It's just been destroyed by the disease. And then finally, and I think this is a really exciting area as well. So again, not about diagnostics, but about understanding the effect of the drugs that you're giving your patients on the brain, is you can start understanding the dosing of the drugs that you're giving. So this again is a tracer that's added for the dopamine system, and this is a person they've had no drug yet. I don't know what antipsychotic they were being given, but then there was a 2 milligram. You see there's a slight decrease in the dopamine receptor availability for this tracer. 5 milligrams less so, and then completely gone at 30 milligrams, and the person may be showing side effects of having their dopamine system completely blocked by whatever drug they've been given. So that's quite a whirlwind tour. I wanted to just give you a taster really. 15 minutes is not enough to go into these techniques in more detail. I hope you've managed to note down the references I've given you. If not, then please contact me on my UCL email address, and know that we have what is a PET MRI hybrid scanner based at the Cancer Centre on Huntley Street, where you can do both MRI and PET imaging at the same time. So thank you very much for your attention. I don't know if we're taking questions now or later.
Yes, so we'll do questions now if anyone has any.
Do you believe that as the imaging techniques are getting better and better, and more sophisticated, we will be able to see patients before they start having symptoms of schizophrenia? Are we quite there yet? Or?
We're not quite there yet, but there is a huge field. Actually, I didn't put that in my talk. It didn't quite fit right. I have done some work where we've been looking at people who've been identified as having the genes for risk of schizophrenia, and we've been looking at structural differences in that cohort compared to people who don't have those genes. Trying to marry genetics and imaging together is supremely complicated because both have very sophisticated modelling techniques to pull out the important parts of the genes and also for the imaging. The group that I used to work with in Cambridge are still working hard on that and looking at a lot of genetics and fMRI and also looking at first-degree relatives as well as being at risk, and trying to understand if there's a sort of a spectrum of these networks before you get to the final marker, if you like. But I do, we're on the right road, but I still think we've got a long way to go.
Thank you.
One question then? Take over.
about genes and their effect on the regulation of glutamate and glutamate.
I wish I could answer that question. I'm afraid I'm not worthy. When maybe one of the other speakers can talk me to that point.
Sorry, do you mind sharing with us like what are some of the constraints with current neuroimaging techniques in detecting schizophrenia?
Yes, I can give you some broad examples. So the effects we're looking for are incredibly subtle. So you've got small effect size, small groups. You've got imaging techniques that are prone to just biological noise, technical noise, and what you're trying to do is manipulate the data in such a way to get rid of all of the background and just leave behind the important bit. And then on top of that, I think schizophrenia is a syndrome rather than a specific disease, and I think that if you can't clinically categorise your cohort well, then it's very difficult for the imaging to pull it apart, because we're still at the beginning of trying to find the biomarker in the first place. On the emission tomography side, the limitation is the time it takes to develop a tracer. It's almost equivalent to the time it takes to develop a new drug, unfortunately. Well, fortunately or unfortunately, I know it's all about protecting the patient, but we're talking about such tiny, tiny amounts of radioactivity and trace amounts of the pharmaceutical that to have to go through the same toxicology as a treatment drug is, I think, quite draconian. But that's my personal opinion.
So, sorry, just a follow-up. So do you think that neuroimaging techniques in detecting schizophrenia, it has to wait until the clinical side for them to have a better definition for the symptom first?
No, not at all. I mean, I think I showed the fantastic use you can make of imaging and understanding how the drugs are working, and you know, some of the big pharmaceutical companies do these things in parallel in order to understand their drug process.
With the NAA imaging, is that showing like metabolism of an area or radioactivity? Like what are you measuring with your function?
Oh, right, okay, so that was an MRI technique, and if I've got enough time, I'll just very quickly try and give you a basic idea. So what happens when you are in an MRI scanner, what we're doing with the radio waves is manipulating the single protons, i.e. hydrogen atoms, in your body in such a way as to manipulate their spins, so that when they relax back out of where we put them, it sends out a little radio signal. And the frequency of that radio signal is very dependent on the chemical environment in which they're in. So most of it is water, and that's why we get these beautiful images. There, you can still detect small amounts that are in other molecules, like the NAA, and so you're detecting the number of protons that are in that molecule, and therefore, consequently, the amount of molecule in that area of the brain that you're looking at. Does that make sense?
Yeah.
Any more questions? Yeah. Okay.
Yeah, I noticed on one of your slides you had, I think it was looking at the dopamine receptors. I think you had N values of 2. I was just saying, how could that be significant?
N value of 2.
I think that's what I saw.
It was the dosage one. There was
Yeah, except the basic.
when it was the was it the when you went?
Oh, it was just showing. Oh, I think it was just showing an example. It was just an example of the fact that when you dose an individual, it's big enough that you will see the decrease in the dopamine receptors available. That's very profound. You'll see that just in a single person. You don't need group statistics to do that.
Okay. One last question? Anyone has one? No? Okay, thank you very much.
Thank you.
Okay, thank you. Thanks very much for inviting me. So the slides are dark, so you're all going to go to sleep now. I'll try and keep you entertained. I'm going to talk, and this is a bit of a follow-on from some of what Anna was talking in the last section of her talk, about this idea in schizophrenia research that's been around for about 10 years, which is the aberrant salience hypothesis. And this has been proposed to provide a link between some of the neurochemical explanations for the disorder and some of the cognitive ideas for disorder and some of the phenomenology as well. So I'll take you through a bit of work that we've done in this area. So it's going to be quite specific, but we'll cover some broader themes as well. And I would also echo Anna's point here that when it comes to mental health disorders, it's true for everything, including schizophrenia, but my own area of research currently is depression, it's true for anxiety as well. What you're talking about is syndromes of symptoms. So you can't assume that these are biologically distinct entities, that a person who has what we call schizophrenia is somehow the same as another person who we say has schizophrenia in terms of what's going on inside their brain. So you could argue that we'll never be able to diagnose, and I would argue that, schizophrenia or depression or whatever it is with a brain scan, because those brains in different individuals might look quite different, even though they give rise to the same symptoms. So I think it's important to bear that in mind, and that, I would say that's a reasonable summary of what a lot of people in this field think right now. So before I go on to talk about what aberrant salience is, I'd better tell you what normal salience is. So salience is a very simple property in psychology, which is the idea that it's a property of an object that makes it stand out from its surroundings and automatically grab your attention, and some people would argue motivate your behaviour as well. So it could be a number of different things that cause this. It could be something like this, like simple feature contrast. If you're going and looking for apples in the tree, then a red object is salient on a green background, visual salience. Or it could just be something that happens very suddenly and you don't really expect it, like this mother playing peekaboo with the baby who doesn't expect to see the face reappear every time. So that's not a form of salience. And the type that I've been interested in is this type, which is things that are salient because they are associated with things that are either have a strong emotional relevance or because they've got associations with rewards. So good examples of this might be something like a fearful face, or a horrible spider for the arachnophobes in the audience. Those of you feeling hungry will find this cake very salient, and of course the ultimate condition reinforcer, of course, is money. So all of every object on here is salient, but the things that I'm talking about are down this end. In fact, I'm going to talk specifically about money because it's an easy kind of thing for psychologists to manipulate in studies. So the idea of salience, in motivational or incentive salience, what we're going to talk about here, originally came from rodent work, and the reason it's important for this aberrant salience hypothesis is it forms the foundation of the link between the psychology and the neurochemistry. So a couple of investigators in the mid-90s published a series of papers, this is Berridge and Robinson, showing that reward is made up of distinct elements, so there might be a preparatory phase, an appetitive phase as they call it, or wanting, versus the consummatory phase, or what they call liking. And they showed that different neurochemicals mediate these two different processes. In particular, dopamine doesn't appear to have much to do with actually eating the cake and enjoying it. It probably has a lot to do with anticipating, getting excited about the cake. So it's this idea of a cue that then predicts something else that's going to happen later on. So that's what they call wanting. And what they showed very clearly is that amphetamine can change this wanting. I'm not going to go into this slide, but what you can see is that you get a marked increase in responding with the amphetamine injection during this wanting period. And other people have argued that doping plays a role more generally in coding salient events. It turns out that salience can go wrong. So sometimes we think things are important in an environment, but they're actually not very important. And Skinner was one of the first people to demonstrate this in the 40s. So he called this superstitious conditioning. He would give pigeons a series of reinforcements, and this would be completely random. He noticed over time that the pigeons would start to develop the behaviours that they'd been doing immediately before they were given that reinforcement. So let's say they've been standing in one corner of the cage or standing on one leg or something like that. They would learn to express these behaviours even though they have nothing to do with the reward schedule. So this is when salience goes wrong, if you like. And this can happen in animals too. So we can see here's, for example, another experiment done with this same salience paradigm as on the previous slide. And you can increase responses on both the reinforce lever, which is adaptive, because that's where you're supposed to be responding, but also on the other levers as well in the experiment. And we can see similar things if we do brain imaging in humans with amphetamine administration, changing the specificity. So salience can be adaptive, but it could also be maladaptive and bring your attention to the wrong kinds of things in the environment. So this is what inspired, along with another other theoretical ideas, Shitesh Kapoor, used to be in Canada, now Dean of the Institute of Psychiatry here in London, to propose what he called the aberrant salience hypothesis. And it's a nice piece of work, and it's captured a lot of attention because it provides this link between the neurochemistry, the cognitive, and the phenomenological. So what Kapoor says is, as a starting point, and based on some of the kinds of data that Anna was showing in the last talk, we've got this very reliable finding of some kind of aberrant regulation of dopamine signaling. And we know from antipsychotic drugs that if you block the dopamine system, you can make some psychotic symptoms go away. Not all of them, but certainly some of the positive ones. So then we have this idea that dopamine somehow might cause schizophrenia, but there's a massive explanatory gap there from going to my D2 receptor to understanding why I think the FBI are out to get me. And so what Kapoor is trying to do is put in the middle parts of that gap, and for that you need a few cognitive ideas. So what he suggests is that you've got this dysregulated dopamine firing, and if dopamine is mediating salience, then you start to think that all sorts of things in your environment are potentially salient or important, so you start paying attention to the wrong kinds of things. And this might be all very well for a couple of minutes, but imagine being bombarded with this constant salient signals for 6 months or a year of your life, which is fairly typical in people who are going to develop schizophrenia, develop a psychotic episode. You come up with this elaborate explanation for what's happening here. You know, I'm constantly being bombarded with this idea that this thing's important, that thing's important. I start to get ideas about maybe people are controlling my brain, and then that eventually crystallizes into some kind of delusion. But that occurs over several months, if not years. And it's not actually until people exhibit some kind of behaviour that brings them to the attention of normally accident and emergency, because they've gone on expressing kind of very bizarre behaviour or done something very unusual. The psychiatrist really gets involved, but that's the end of the process. At the beginning of the process, the dopamine dysregulation started much earlier, probably all the way through their life, in fact. And then he proposed that we give them antipsychotics, which block, if you remember, the postsynaptic dopamine receptors, as I'm explaining, but they don't change the regulations. They don't change the fact that the dopamine itself is still being released in a dysregulated manner. And so Kapoor proposes that the antipsychotics might also dampen the positive or adaptive motivational effects of abnormal events. But in particular, this hasn't taken away the underlying problem. If you like, it's treated the consequence of the problem, not the cause of the problem. And eventually, because of these side effects, almost all patients eventually stop taking antipsychotic medication. And at that point, if they've been on their drugs for a long time, then there may be some kind of extinction or unlearning of these associations that have happened here. But as soon as they stop taking the medication, then the aberrant salient signals start to get through again, and then that leads back into the spiral here. So that's Kapoor's explanation for how aberrant salience causes psychotic symptoms. So if you like, it's an extension or a cognitive elaboration of the dopamine hypothesis. So I'm aware that I'm speaking a bit fast here, but I'm going to try and get through everything. So I just want to check whether everyone is happy with the model, because this is going to form the basis of the rest of the talk. Okay. So at the time when Kapoor proposed this, it was a great idea, and the problem was there's absolutely no evidence for it at all. So what we set out to do is to try and provide some of that evidence. So we invented a very simple test, very artificial, laboratory, abstract, if you like, but with the intention of trying to tap into this motivational salience process, both the normal or adaptive motivational salience when salience is helping you out, if you like, in the world, and aberrant salience that you've assigned to something that shouldn't really have salience assigned to it at all. And we thought we would design a task to look at this, very simple task. And this is what we came up with, and we call it the salience attribution test, very original naming. And what we do is we present a cue here, and then the subject is going to see a black square, which is going to appear very fast, and all they've got to do is press the square as quickly as they can, and then they'll get some feedback, and sometimes they're going to win some money, and sometimes they're not going to win some money. And it's as simple as that. And there are 4 different types of cues, and they've each got 2 dimensions in them. One's a colour, and one is a form. So the colour, blue or red, might indicate whether you're likely to get a reward or not. So it may be that the blue ones are very likely to give you a reward, and the red ones are unlikely to give you a reward. And you don't know anything about this at the beginning of the task, you're just learning by trial and error. And then there's this other dimension as well, which is the form dimension or the category, so the animals or the household objects here, and they might be completely irrelevant to whether you're going to get a reward or not. So 50% of the time if you see an animal like a kangaroo, you're going to get a reward. 50% of the time when you see a household object like an iron here, you're going to get a reward. And these 2 dimensions allow us to calculate these 2 different measures of salience, we think. The adaptive measure of salience, which is whether you get quicker to the blue stimuli than the red stimuli here, so that's our implicit measure, and the rating that you'll give to how likely, at the end of the experiment, how likely you are to get a reward from following the different types of cues. So if you show high adaptive salience, you'll respond more quickly here to the blue than the red, and you'll rate the blue ones higher than the red. But if you're if you're a computer playing this game perfectly in a Bayes optimal manner, then what you would do is soon hone in on this task-relevant dimension here. You'd soon learn that the blue is good, the red is bad, and the animal versus household objects basically doesn't make any difference. But let's say your learning system is not optimal. Let's say you've got some kind of aberrant dopamine signaling going on. You might spuriously learn that, for example, the animals are actually better than the household objects, even though in reality they're not. Subjectively, that's how it may appear to you. So we can measure the differences along the other dimension to give us an idea of the aberrant salience. Okay, so those are our measures in this cognitive paradigm. We've got the adaptive measures and the aberrant measures, and there's an implicit one and an explicit one. And I'm going to talk about this one because that's the one that gave us basically all of the results in our studies. So the first thing that we did was to test a group of people who experienced frank psychotic symptoms. These are people in their first episode of psychosis, and almost all of them are medicated. This is standard for research in schizophrenia. Very hard to test people who are unmedicated who also have acute schizophrenia. The reason being, of course, that there's a clinical imperative to put them on the medication. And so it turns out that people who have these symptoms and are currently medicated show reduced adaptive salience. Remember, that's the difference between the real high and low probability, both in terms of their reaction time and in terms of how much they rate the stimuli as being high probability or low probability. So this is interesting, actually replicated a whole load of other research suggesting that patients with schizophrenia are not very good at learning the contingencies between cues and rewards. What about aberrant salience measures? Remember, this is the one that goes along the irrelevant dimension. So they shouldn't be learning that one is better than the other. So it turns out that even in people who don't have schizophrenia, we're not perfect at learning, display some kind of aberrant salience. That's true if you look at reaction times or if you look at the rating. But much to our disappointment, the patients with schizophrenia didn't seem to show any aberrant salience either. So when we first analysed these data, it was kind of underwhelming, I suppose. But then we took a look at the symptoms, and this was quite exciting. So it turned out that in those who had their delusions well controlled by the medication, and that's assuming they were actually taking their medication. Not all patients with schizophrenia actually take their medication. Important to remember that as well. In fact, not all of ours are taking their medication either. But the people who didn't have the delusions, they score very low on this aberrant salience measure. But the ones who had the delusions are scoring relatively high, higher than the healthy control group. And we also, it doesn't matter if you split this as delusions or no delusions or a continuous measure, the severity or the intensity of the delusions. You see this relationship with this aberrant salience measure. So that was quite exciting because that was really the first, I guess, behavioural evidence that was inspired by the aberrant salience hypothesis. And this, of course, corroborates the hypothesis, though not in the complete sense that I'm sure Kapil would have liked us to show. But this is the first experiment that we did to show that. And there's also an association with something called schizotypy in healthy volunteers, which is a personality trait which is related to schizophrenia. And it's important to remember this because, of course, schizophrenia, depression, anxiety, these things don't live as disorders in the real world. They really live on spectrums, and that's what we're seeing here in these data as well. So the next experiment we did used a bit of a different approach. We're using the same kind of task, hi Rick, and the same idea. So we're looking at people who've got psychotic symptoms, but we had an unusual opportunity here in testing people who are very likely to go on to develop schizophrenia, and they've already got some attenuated psychotic symptoms, as they're called. So sometimes we think of these people as in a prodrome for schizophrenia, a period of time immediately before the frank psychotic episode. But they're not actually yet ill. In fact, they could easily be coming to take part in your psychology experiments, psychology undergraduate students. You wouldn't be able to differentiate them necessarily from a healthy volunteer group. But if you ask them the right kinds of questions, they might admit, for example, to having some very suspicious ideas or very occasional perceptual abnormalities or maybe minor hallucinations. And so we call these at-risk mental state subjects here. I'm very sorry that this pointer has come off. It's probably another one here. Never mind. So that's the at-risk mental state subjects there. And again, we're looking at these aberrant and adaptive salience contrasts, this time both for behaviour and using functional magnetic resonance imaging to have a look at what's going on in the brain in the same way as Anna describes in her talk. And so this is what aberrant salience looks like in the at-risk mental state. A bit of a different picture to what we see in medicated first episode patients with schizophrenia. Here we see across the whole group, let's come back again, there's an overall elevation in our behavioural aberrant salience score in relation to the healthy volunteer sample. Neither of these samples are medicated, which is probably very important. These are the same data expressed in two different ways. But interestingly, this adaptive salience at this stage in the disorder appears to be relatively normal. So they're showing the regular, appropriate salience, if you like, but they do seem to have this slightly increased aberrant salience. And it turned out that this correlates with the symptoms. So you can see this is actually with the personality measure that I was talking about before, schizotypy. And if we look at their positive delusion-like symptoms, I should say, because none of them have got complete delusions as yet, but they're certainly on their way, some of them, to developing delusions, you see an association with those kinds of symptoms as well. That's depicted here. And then with the functional magnetic resonance imaging, we can also look at the salience responses inside the brain. So when we run a contrast of, if you think of high versus low, they're not truly high versus low probability contrasts, but they are subjectively to the participant. So they believe that they're, one, for example, the animals are high probability and household objects are low probability. You see differential responses in this region of the brain, the dorsolateral prefrontal cortex, which has been highlighted in a lot of other studies, some probably from some of Anna's former colleagues in Cambridge, that have used these kinds of associative learning paradigms as operating abnormally in this kind of context. And if we look in the ventral striatum, which is the target of, or a strong target of the brain's dopamine neurons, we see also changes in both the aberrant salience contrast, that's here at the top, and the adaptive salience contrast here at the bottom. Interestingly, it turns out that adaptive salience signals in the striatum look relatively normal in people who have these attenuated psychotic symptoms. But the relationship between the degree of the striatal response here, and make sure I get the right one now, yeah, that's this figure down here. The striatal response down here and the degree to which they learned inappropriately was different in the people who have these attenuated psychotic symptoms. In fact, it was uncoupled with the responses in the striatum. And we also showed an association between the degree of response in the striatum and these kinds of delusion-like symptoms here. So I've been talking about salience attribution, but I wonder how many of you know about other theories of what dopamine does in the brain. So put your hand up if you've ever seen this figure before. No? Okay. Oh, one or two. That's great. So you're doing computational neuroscience, of course, which is good for you. So this is an idea about what dopamine might actually be doing in the brain. It sort of, if you like, incorporates a bit of motivational salience as well. But what this idea says is that the dopamine system acts as a very simple type of learning mechanism in the brain, using a quantity called the prediction error. And what this account says is that dopamine signals prediction errors. And it's very simple, a prediction error. A prediction error is the difference between what you thought you were going to get and what you actually got. So let's say you're going along and then you get a call and someone tells you you've just won lottery. That's going to be a positive prediction error because you had no idea that was coming. And that's probably going to give you a little burst of dopamine, like this monkey here who's getting an unexpected juice. But let's say instead that, let's say you're going to work every day, you know you're going to get paid at the end of the month, the check comes through or goes into your bank account, you look at your bank statement, it's pretty dull. And that's the kind of thing that's happening here when the reward is fully predicted. And then let's say you're expecting to get paid at the end of the month, and unfortunately the university finance people have gone on strike and they forget to put the money into your account, and then you're not going to get them, you're going to get a negative prediction error like this. And that's the idea of prediction error. It's a learning signal. Better than expected, as expected, or not as good as expected. It's very simple. And it seems that dopamine seems to encode this in the brain. So if you look at signals in dopamine neurons and how they correspond on these kinds of learning tasks, they seem to follow this kind of pattern. This is a really important idea in the field of computational psychiatry, which is a new way of trying to understand how we think the brain is operating in mental health problems. So, if dopamine is dysregulated, different kinds of things might be happening in this system. So, for example, we might have this scenario here when the reward's predicted and the reward occurs, or the reward's predicted and no reward occurs, and you can see the different patterns of response. But if the dopamine's firing abnormally, you might get something that looks like this. This is what Kapoor's talking about, the idea that all of a sudden there's this burst of dopamine which is giving you this prediction error, and that's what's actually driving this aberrant salience. But there are other possibilities as well. It could be that this dopamine system is, for example, filling in the gaps here, so you might be bad at learning from omissions of reward, or even that the dopamine system is just firing all the time and you have no way of distinguishing between the appropriate and the inappropriate salient signals. So I just put this in to give you an idea of where the field is going in terms of trying to understand how these neurochemical and cognitive ideas might map onto computational terms as well. So I'm just going to leave it there. I'd like to thank lots of collaborators, and of course, thank you very much for your attention. Sorry if I went over time. I feel like I should put the lights on at this stage.
Any questions from the audience?
So does the aberrant salience theory, does it explain negative symptoms of?
Well, it might do, yeah, it might do. So, for example, let's say you've got a system like this, and let's say that the tonic activity is very high, so the baseline activity is very high. That might make it very difficult to pick out an adaptive salient signal, like a reward prediction error, from the background noise. So that means even when something good happens, you don't really perceive it as good, or your voting system doesn't process it as being as good as it should be. So if that's what your experience is, then you'll end up not attributing as much motivational salience to that particular stimulus. You won't get as excited about going out to see your friends or have dinner or whatever it is than you would if your dopamine system was operating normally. So I think there is a potential explanation for negative symptoms as well as positive symptoms in this framework. But that's quite speculative at this stage.
Do you find that aberrant salience occurs in other disorders, which is in psychosis?
Yes, so there's been one study done using our task in people who have Parkinson's disease, and I don't know if anybody in the audience has had a relative who's had Parkinson's disease, but you tend to take medication that targets the dopamine system, and in some people that can actually cause the dopamine system to go awry, much like in psychosis. So if you look in people who have, if you like, too much Parkinson's disease medication, there's been one study using this task to show that they also show elevated aberrant salience, which has provided some convergence findings supporting that. That's a very good question.
Just following on this question, would you find the same kind of effects, results in other kind of psychiatric disorders, like, I don't know, bipolar disorder, for example, which also show psychosis as a symptom?
It's a prediction, yeah, it's a prediction that would be made. So again, it's come back to this idea that we don't really have categorical disorders in nature. To quote the famous phrase from Newton, we're not carving nature at its joints when we do psychiatric classification on the basis of symptoms. We probably have something along the lines of a spectrum of things that can go wrong with the dopamine system, of which this type is one, and that might be common to lots of different disorders. So you're right, there's psychosis in lots of different scenarios, and one would predict that you would see that in bipolar as well, maybe also in people with Alzheimer's and also have hallucinations and delusions. That hasn't been tested yet, so I can't tell you.
Any more questions? Yep.
I was just wondering if you know of any studies or reviews where the subjects are grouped according to their symptom profile instead of the disorder that they've been diagnosed with.
Yeah, so that's very common in what people call analog samples. So this would be taking a spectrum approach to understanding the disorder. So typically done in large cohorts of university students. You know, you get 200 or 300 people, get them all to fill in a questionnaire, like a schizotypy questionnaire. In fact, Dr. Adams over here is going to talk exactly about one of these kinds of studies that he's done in a few minutes, so I won't talk too much about it. This doesn't work completely brilliantly for psychosis, which is quite an extreme state, but things like anxiety and depression, which are very common in the population, the spectrum goes a lot further down into what you might consider to be normality. And so yes, that's a very common approach, and it's also an approach that's going to become increasingly common as people start to, especially scientists, start to do away with the idea of just looking at the classification. And there are some political changes that have happened in America which are going to accelerate that, and people stopping funding studies that just use these categorical boundaries. So that's very much the field, the direction the field is moving in, yes.
Question back?
In very simplistic terms, if you say that basically there's a stimulus and there's a dopamine response in the brain to that stimulus, if that dopamine response occurs without there actually being a stimulus from the outside, is that maybe a sign that the wiring of the brain could be more of a two-way street, that could actually cause the brain to think that there is a stimulus that isn't there in the first place?
Well, the dopamine, so when you experience a dopamine, a burst of dopamine, we don't really know what that means. We can't measure that in humans because we don't have the kind of imaging capabilities to be able to do that in real time. You could measure that in animals, but you can't ask them what they experienced. So you sort of have a bit of a catch-22 scenario. Most people think that when you get this dopamine firing, it's tied in some way to some kind of external event. Could be tied to some kind of internal event, of course, and something that you might be thinking about at the time, and then that takes on a great deal of importance. So I think the idea is that this, in the Kapur theory at least, the aberrant salience hypothesis is that the dopamine, the dysregulated dopamine or inappropriate, contextually inappropriate dopamine firing happens and it tags something. Even if it happens in isolation, it might tag an internal representation, an external stimulus, but it imbues somehow that representation or stimulus with increased relevance and tells you to pay attention to it, tells you maybe to work towards it. And Kapura's arguing that's what's driving particularly delusional type symptoms where people get suspicious ideas, for example.
So it's dysregulation in response to stimulus.
Yeah, but you could argue that it can also happen in response to internal stimuli. I think that's the way in which he conceptualizes hallucinations, but it's not nearly as well worked out as the idea behind delusions. In fact, he only mentions hallucinations once in that whole paper.
Thank you very much. Okay, thanks everyone. Can everyone here at the back? Okay, my name is Rick Adams. I actually work in Jon's group at the moment, but I'm a clinical lecturer in psychiatry, so I did medical training and been a consultant psychiatrist for a few years. And I'm based partly at the Institute of Cognitive Neuroscience with Jon, and also in the division of psychiatry. And I'm going to talk today about to skirt over some computational approaches to modelling what might actually be going on in the brain in psychiatric disorders, specifically schizophrenia, in computational terms, thinking of the brain actually as a computer. Now it's late, and I'm not going to go into too much maths, so but I think the basic concepts are relatively easy to grasp, and I think they're really interesting. So one of the more interesting things about research into delusions is given the strangeness of some of the things that people who suffer from schizophrenia say, or the strangeness of some of the things that they believe, it's actually been quite difficult to demonstrate significant reasoning abnormalities in that population. So, for example, if you give them logical reasoning tasks, that they seem to be just as good at those as people from the general population. And it was a long time before people actually found reasoning tasks at which they performed differently. And this is probably the most famous one. And since it was discovered around the 80s that their performance was different in this task has been really done to death by the psychological literature. It's been repeated and repeated and repeated because it's one of the few replicable findings. So if you ignore these sequences under here, and just imagine that you've walked into the room and you're shown these two jars by the experimenter. One of them with mainly green beads in and a few yellow ones, and one with mainly yellow beads in and a few green ones. And then the experimenter puts those jars under the table where you can't see them, and then one at a time they just pull out a sequence of beads from one of those jars, and they ask you to just say, when you're sure, which jar it's coming from. Simple as that. And if you imagine seeing this sequence here, where do you think most people would say, okay, I think this is the green jar? Shout out some numbers.
4?
Yeah? 4, 5, 3, 4, 5, exactly. Yeah. So if you look at, this is the bead that people pick. So this is a normal population. And 3 and 5 are the kind of peaks here. 4, in this case, is absent just because the bead is a different colour. But were it green, I'm sure there would be a peak here. But you can see in the psychotic sample that half of the people say that they're sure about the colour of the jar on the first bead. So they see one green bead and they say, okay, I've seen enough. You don't need to show me any more beads. I know which jar it is. And there's another variant of this experiment whereby you show a much longer sequence, and instead of just asking them to stop the sequence when they've seen enough, you ask them after every bead, okay, what do you think now? What do you think now? What are the probabilities on like a sliding scale? And this kind of experiment, you get much more data. You get 50 data points as opposed to just 1. So we did this test, and the project I'm about to present was actually a master's student project from last year. So the nice thing about computational projects is that you don't have to learn how to do fMRI in 3 months. You can actually collect the data and analyse it relatively quickly. So here is the sequence we used, and you can see there's a change of jar in the middle, but these beads are just presented one at a time. And this is the sliding scale at which the subjects were asked to indicate which jar they thought it was, whether they were sure or not sure or about blue or red. And I'm not going to go into too many details about the model that we used to model this, but it's worth getting the simple ideas across. So it's a probabilistic model, okay? And what it's modeling at the bottom here is the color of the bead that's being produced. And that variable is given by x1, 0 or 1, basically, for blue or red. And the variable that affects this is this variable here, which is the probability that the jar is red, or the probability that it's blue. So even if you know this with absolute certainty, that you're totally sure that this is the red jar, you're still only 85% certain that the next bead will be red, because there's the distribution of beads within the jars. Okay? And then at the level above that, we have what's known as the volatility, or the probability that the jar might change at any point. So our subjects don't know whether the jar's going to. They know it might change, but they don't know how many times it will change, and they don't know how long the sequence will be. So they're quite unsure about, if they see a blue bead, whether this is a change of jar or just a chance event. And here is the response that they have to give. They just put an arrow on this sliding scale saying how sure they are, and there's a bit of noise injected into the system there. So that's the model on one trial. And all this model does is then move through time, from time step to time step, across 50 time steps, one for each bead. And these values will slowly change. And the amount that they change from trial to trial is determined by what the subject sees and whether he expects what he sees. So as John was talking about before, if the subject has a prediction error, it can adjust his or her beliefs quite a lot. If they see what they expect, they're not going to change their beliefs much at all. So, and the amount that beliefs change from trial to trial is given by a bunch of parameters which I'm not going to go into because I've only got a short space of time. So we have healthy volunteers from our ICN subject pool, about 96 people who all completed these schizotypy questionnaires. Schizotypy is essentially a measure of not even symptoms, but beliefs or experiences that are somewhere akin to being psychotic, but are clearly not psychotic. So these could be feelings of paranoia, these could be feelings that superstitions or beliefs in things like telepathy or feeling controlled by external forces, but vague things like that, nothing outright hallucinatory or psychotic. And the first effect that we talked about, this initial effect we didn't see in the sequence, so I'll just pass straight over that. And but the interesting point is, after the change of jar, we thought that the schizotypal individuals would be very quick to change their beliefs. You know, just like the patients who decide after one bead that they're happy about the jar. But actually, the complete opposite happened. That they actually took quite a long time to change their ideas. And so we asked our model why this was. That's just a graph showing that. And if you imagine, it's all to do with this number here at the top. So this volatility probability at the top. And if you imagine it starts out, if you imagine this graph is just a plot of this number going from the very start of the experiment to the very end of the experiment, from here to here. And it starts at some value where they don't know if the jar will change 5 times, 6 times, only once, they're not sure. But they have a certain amount estimate of that value, and then as this experiment goes on, they see lots and lots of red and lots of blue, and they realise there's only been one change. And so their estimate of the volatility will decline during the experiment until it reaches some sort of flat level, which is what I've kind of sketched out here. And that flat level is given by this M parameter, which is the only important one. And what we found is that the more schizotypal subjects had a lower M parameter, a lower estimate of volatility. What does that mean? So let me show you on these graphs, and you'll get a sense of what's going on. So these 3 levels correspond to those 3 levels in the model that I showed you before. Okay? So at the bottom here, we've got the actual beads that they saw, blue or red, blue, red, 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 red. On the diamond shapes are where they actually put the arrow on the slider, how sure they were about the reds or the blues. And the purple line is our model's fit of their data. So our model's best attempt to reproduce what they did by adjusting its parameters until it gets its performance as close as it can. You can see it gets pretty close. And at the next level up, we have their belief about the jar. So red is up here and blue is up here. And so you can see this person at the start of the experiment sees a mixture of beads and then a bunch of red ones, and ends up quite quickly believing that the jar is red and this is maintained. And at the top level, you have this belief about the volatility. So it starts at a highish level and then declines and declines and then reaches this kind of asymptote where it just stays towards the end of the experiment. And the thing to note about this is that when the evidence changes, that this person can adjust their beliefs quite quickly from one jar to the other. And the reason that person can adjust their beliefs quickly is because at this second level, you see these confidence intervals around this estimate of the probability of the jar here, shaded in green. So what these confidence intervals mean is that although the subject thinks the jar is red at the moment, he thinks it might change at any time. That's the point. Because his estimate of volatility hasn't gone down that much. But if his estimate of volatility goes down further, he's going to be more confident about the jar, but because he's more confident about it, he's going to be less willing to change his beliefs about it. And that's what you see if we look at a highly schizotypal subject. This person takes a long time to make up their mind, and an even longer time to change their mind when the evidence changes. You can look, only at this point do they say that they're certain about the jar after seeing 10 blue beads in a row. And the reason for that is because this volatility parameter here has come down quite quickly to a low level, and it's shrunk these confidence intervals about the identity of the jar and made it hard for the person to update their beliefs. Okay? And at the moment, we're currently investigating whether there is a dopaminergic basis to this parameter, but that's some other work that I'm not going to go into now. So, to give a kind of psychological spin or view on this, we looked in the literature and found this experiment 10 years ago this guy had found that this psychological construct called the need for closure had been something that people had looked for in order to explain psychological reasons for delusions. So perhaps the idea is that people who become deluded are intolerant of uncertainty or ambiguity. So they have these strange experiences, and not knowing what is behind them is worse than thinking that NY5 is behind them. Or having an assassin in the shadows is worse than having an assassin with a real face, even if that face you've put on him. And so when they look for need for closure or intolerance of ambiguity, they have found that it correlates positively with aspects of delusion prodromus. But the thing that puzzled him was that decisiveness seemed to correlate negatively with intolerance of ambiguity. And he was puzzled by this because he thought, well, if you're intolerant of ambiguity and you want to make your mind up about something, why should that make you indecisive in real life? Well, this makes sense here because what it allows you to do is make your mind up, but then if the evidence changes, you can't change your mind. So making decisions is actually hard when you're intolerant of ambiguity, not easy. So that's just one small point. That was a master's project, so it did very well. I was very pleased with that student. And if you're interested in these kind of approaches, if you're interested in other kinds of disorders other than schizophrenia and stuff, we have a computational psychiatry course just started last year at UCL that is going on in April, and you can Google it, or if you've got time, you can try and write that down in the next 30 seconds. And it's free to people at UCL, so you can just come along and see a bit more detail. And if you're interested in reading about it, there's a review written by 4 of the top computational psychiatrists in the world who are all at UCL in Trends in Cognitive Neurosciences last year. So I just thank all my collaborators and leave it there. Thanks.
So any questions from the audience?
Yeah, to what extent can the work and others like this be reconciled with more classical psychiatric approaches like psychopathology or analysis? Or is that all actually thrown out the window?
No, so one really interesting thing I've noticed is that these are the collaborators that worked on this study with me. So you see Jon sat here in front of you, but this was my master's student, and Christoph, Michael, and Carl are all people who have either physics degrees or a huge amount of knowledge of physics and statistics and mathematics. But they all have done psychoanalytic training early in their careers. And I think at first when I discovered this, I was totally astounded, because you think that these two approaches are incommensurate. But actually, the reason is, I think, that they were all in their own way kind of unsatisfied with pure biological explanations of mental disorder, but not really knowing where else to look and investigate. And Michael in particular has been working on actually trying to make computational models of interacting minds, so people who are trying to do a task like this, for example, but predict someone else's behaviour and understand how someone else will do it. And in this review article, they discuss how to model disorders like autism and personality disorders where people have difficulties understanding other minds or react abnormally to other minds. So actually, all disorders and approaches are amenable to this kind of analysis. And it just, the good thing about this method is it quantifies it and makes it much more experimentally testable.
Excellent. Any other questions?
Yeah.
I don't know if you can answer to this question, but if you were to repeat the same exact experiment but put a factor of a reward, so let's say they would get like a positive reward or a negative reward, again maybe using money, like they would be detracted certain money, do you think that the results in the schizophrenia, in schizotypy, would change?
It depends how big the reward is or how much you punish people for making mistakes. Because if you do these beads tasks and you give people a pot of money which they lose if they say the wrong bead, then this jumping to conclusions effect of choosing on the first bead disappears. So
even in schizophrenia?
Yeah, even in, because and it makes sense, because there's a lot of money at stake. But nevertheless, if you don't completely skew the results with a huge reward, but if you just had a rate of 5p type thing all the way through, I don't think there would be. I don't think there is a fundamentally different inferential process to what is between a reward learning paradigm and something that ostensibly doesn't have reward. Because you can argue doing an experiment and getting it right is its own reward, and for other complex reasons, I don't think there's probably that much difference between the two.
Okay, thank you.
Anything else? Okay, thank you very much.
So my name is Muj Husain. I am a psychiatrist, and I'm not from UCL. I'm from across the river, so I hope you don't think that's the enemy. So I'm year 6 in my kind of psychiatric career, which means I'm right at the cusp of being a consultant. I have been in psychiatry because I've done kind of time out doing other things for roughly 8 years, and I work in Lambeth. I've worked in South London for my whole career. I'm going to try to tell you what it's like to be a psychiatrist, hopefully giving you a sense of where this research is, what it's doing kind of in practice. I realise I have no idea who all of you are. I'm guessing you're a mixture of neuroscientists and psychology students, is that right?
Yeah.
Anyone else? Anyone different?
Pharmacology.
Pharmacology. And
Professional arts.
what's that, sorry?
Sort of liberal arts.
Okay. Excellent. Okay, so I will try my best to pitch it at what I think, what my guess would be a reasonable level. If it's way too basic, just pitch in and it will devolve into a free-form discussion. I'm very happy with that. Okay, so I'll tell you a little bit about what we do, about what my work involves at the moment, a little bit about psychosis and schizophrenia. I get a sense you'll probably know most of what I'm telling you there. The kind of approach we use to diagnose and treat people and working together on that. And I'll give you a couple of cases because I think it's always more interesting when you're hearing about real people. I'll end with some of the challenges that we face in the profession and why I still work in psychiatry. Before I do that, in preparation for this, I thought it would make sense for me to do a Google to see what the general flavour and impression is of what psychiatrists do, just to give me a sense of what level I might pitch this at. This is what I found. I thought there would be at least one good thing, but no. Yeah, the very last one, evil. So, I think, I found this and I had to laugh at it, and clearly we're laughing at it here. I think it represents something kind of deeper. I think it's something about the stigma against mental illness that gets transferred to psychiatrists. I think there's something going on with why this comes up on people. And I'll come back to that later on. Okay, so what do? We work in large teams, so we are not the people always making the decisions about patients. We work, it's very much a kind of an approach where we're collaborating with others. So we work with nurses, we work with social workers, we work with psychotherapists. And generally psychiatrists, our jobs are to kind of lead those teams and to bring all those different approaches together to help patients get better. We are, as you pointed out, medically trained, so we're all doctors. We do our kind of foundation doctorate, which is your general medicine and surgery. Then we choose, just like a cardiologist chooses to be a cardiologist, we choose to go into psychiatry. And we're trained in prescribing and we get training in psychotherapy. And our job is to bring those approaches together. There are several branches of psychiatry. So I've listed some of them there, those are the main ones, but I think that's something that people don't always know, that there are so many different types of psychiatrists out there. Right, on to what I do. So that's Lambeth Hospital on the one side. It's a particularly sunny day, it's often much grimmer than that. But that's where I do a split job. I work partly in the hospital and partly out in the community. That's Brixton. So my hospital job involves working on an acute ward. I work on a ward where every new patient in the borough who's just been admitted comes to our ward, and we have to make a decision over 7 or so days as to where they're going to go. Are they going to be able to go home? Do they have to be on a longer stay ward? So it's really kind of rapid decision making. And the team I work with in the community is a home treatment team, which is wonderful because I get to see people in their home environments. You get so much more from that than you do when someone's been seen in kind of like a psychiatric office. You learn so much more about the people you're working with. So those are my 2 roles. And actually, while I'm here, I think it's worth pointing out that Lambeth, like most areas of London, including probably certainly up here, it's an area where there's a lot of social deprivation, really high drug use, lots of migration, particularly with kind of Tommy's just St Thomas's Hospital near Westminster. around the corner. Everyone comes into Waterloo from Europe, so we see people who've just come in. Often people with kind of psychotic disorders will just decide to kind of get on the train and go somewhere else. So we see a lot of that. So there's really high levels of psychosis. And I don't know if this is still true, but in Camberwell, which is just next door to us, it was once the kind of highest rates of schizophrenia in Europe. So it's really very high levels. And that plays out on the wards. We see huge amounts of psychosis and schizophrenia. In fact, I think it's probably the mainstay of what we do, for better or worse, at the moment. Right. So what is psychosis? I think this is probably all kind of bread and butter to you. It's a group of symptoms. So psychosis is not a diagnosis. And the kind of main symptoms that we're looking for are delusions and hallucinations. Delusions being thoughts, hence I've got that the wrong way around. Well, it would have been amazing, but I'll go and work on my slides. But does anyone know why this one is in, or why hallucinations would have been in this speech bubble rather than something else?
They might be auditory.
Yeah, they might be auditory.
And they're more real, are they?
Sorry?
They're more real.
Yeah, they're absolutely. I mean, to the person, they're absolutely real, and I think that's really crucial. They are as real as the ground being under my feet or the sky being above me. There is no kind of persuading someone out of it, particularly when they are acutely unwell. We sometimes challenge it to work out, not to try to persuade them that they're wrong, but to work out whether this is a delusion or whether there may be something else going on. But yeah, it's absolutely real to them. The reason why it would have been a speech bubble if I'd got the things the right way around is because in schizophrenia, it's usually auditory hallucinations that we see. We do see, I think there's a whole range of things that can happen, but we do see kind of visual or olfactory, so smells, tastes, being presenting as hallucinations. But our thinking there is that we have to rule out other causes if that happens. It's much more likely to be what we might call an organic cause, so something kind of obviously wrong in the brain which isn't related to just schizophrenia, so you'd be doing lots of other medical tests in those cases. And yeah, I think the essence of it is a loss of contact with reality, and that's what's happening with both delusions and hallucinations. People become quite disordered in their thoughts. So what we mean by thought disorder is the connections between thoughts start to kind of disintegrate in a way. So you'll be jumping from one thing to another, and it becomes increasingly hard to make sense of what someone's saying. The thoughts might be, sometimes some types of psychosis, they might be kind of really sped up, so it's going from one thing to another really quickly. Sometimes you see it really kind of slowed down. And frequently we see, well actually not that frequently, but we do see kind of thought interference, where people get really unusual experiences, where they feel that thoughts are being put into their heads somehow, or thoughts are being withdrawn, or that thoughts might be kind of spread out to other people, being broadcast somehow. It can often be part of a kind of a larger delusional framework. And obviously, if people are kind of believing and disordered in their thoughts in that way, it will have an impact on their behaviour, and that's what we see. Okay, so on to schizophrenia. So I don't have any bullet points here, I'll just talk about it, but this is a painting by William Kurenek. He is actually, there's a painting of his up in Lambeth Hospital as well, not this one. But he's a Canadian artist. He was treated at the Maudsley, which is one of the places where I work. And he's spent a lot of his career trying to depict schizophrenia. I think it's probably quite hard to depict, which is what this demonstrates. So it's well worth kind of Googling him, I should have put his name. But he does wonderful paintings. So schizophrenia, it's one of the ways it presents is psychosis. In fact, the mainstay of what we're looking for are those psychotic symptoms. But it's where those psychotic symptoms, one, don't have another cause that could be contributing to them, and also there's a chronicity to them. They're kind of long-term symptoms. It becomes like a relapsing, remitting condition, a bit like multiple sclerosis, where people get better, and then they're well for a period, and people then relapse. There are lots of different courses that it can take. So some of them are kind of relapsing. Some people will very rarely be symptom-free. So you can get a whole range of ways that people present. Typically, the kind of patients that I see in Lambeth, the most common are, I guess, young men presenting, and we know schizophrenia presents in kind of late teens, early 20s, most commonly for men, a little bit later with women. They usually present in my line of work because we're kind of an acute service, because they're doing something which people are worried about. So it's very rare that someone comes themselves and says, I think I've got schizophrenia or I might have psychosis. But sometimes people do come to A&E, say, people are spying on me, can you help? Or things like that. So, but often it's families or other services like housing or the police who are bringing people in. Before that kind of onset, before they come to me in my acute service, it was referred to earlier on, that kind of prodromal phase of the illness. People are often slightly unwell for quite some time beforehand. And what often happens is people will have what we call a delusional atmosphere. That's where they just have a feeling, they know something is not quite right. They can't put their finger on it, but they just know, they're sure about that. And then one day something will happen, like they will see traffic light changing colour, or they'll see something that has kind of been misplaced in their house, and then they'll be, that's it. Someone's breaking into my house and moving stuff. That's why I've been feeling this way. And they'll start to then put these kind of connections together. And I guess hearing about salience earlier on, they'll start to kind of make connections, probably getting those kind of dopamine hits as they're doing so. And it all kind of comes together to form a delusional kind of wider framework. So that's what we see. And then I guess the other end of the spectrum, we see a lot of people who've gone past that kind of acute psychotic stage, and I think someone mentioned negative symptoms earlier on. We see people who have a kind of a lack of motivation, a lack of affect, so they don't appear to have any particular emotions, often kind of poverty of speech, they're not really saying very much. And that often happens, it can happen at any stage, but it often happens kind of later in the illness. I think one thing, it may have been mentioned in some of the research talks you had, but one thing to highlight is that we know that schizophrenia is not one condition. Most likely there are several different pathological things going on, and we're only just starting to piece together. As we heard, we group it as one because it responds to the same treatments sometimes, and some of the symptoms go together. But this is why when I'm seeing people with schizophrenia, they present in a really kind of wide variety and wide range of ways. I think that reflects the fact that there are different processes probably going on ultimately, and we're learning about that. Right, so how do we assess and treat people with schizophrenia? Has anyone come across this model before? Is this familiar stuff? Yeah. So you've heard, I think, lots about the biological stuff. Our job in psychiatry is to bring three things together: the biological, social, and psychological factors. So when we're diagnosing people, and kind of assessing people, I'll be taking a history, kind of spending an hour or so, or more sometimes with people. I will be trying to work out which of these kind of risk factors and things are present in their history and their story. And I will be looking to make a diagnosis, so to put a label like this might be a psychosis or it might be schizophrenia. I'll probably have a differential diagnosis where I say these are the other things it could be, but I think those are less likely because. But in psychiatry, unlike I think in other medical disciplines, we also have a second element to our diagnosis, which is what we call a formulation. And that's because we recognise the diagnostic labels are not really sufficient to capture what's going on. So diagnosis is a very kind of classificatory way of dealing with things. There's also a kind of a narrative understanding, so the story of what's happening with someone. So we will kind of formulate that in a description of what's going on, and that's just as crucial to our diagnosis. Much harder to capture, I think, in research, but that's really important. And then this model is what we use when we're treating people. I sometimes like to think of it as a bit like kind of a machine when someone comes in with kind of dials, where your job as the kind of admitting team or the team dealing with someone is to turn these dials and tweak them to try to get someone better. And you have to think about potential dials in all of those three categories. So if we just focus on, say, the medication dial, say you take your antipsychotics, we may not be dealing with their kind of family disruption, the stresses that precipitated them relapsing in the first place, or the psychological issues. They may be kind of very low in mood, really needing kind of psychotherapy. As people tend to get better and improve from psychosis, that kind of psychotherapeutic approach becomes more and more important, because that black and white division of people not believing or believing their delusions are 100% real starts to dissolve a little bit. So we, I guess, can kind of get people to understand what their delusions are, what makes them more likely to come on, what makes them less likely to come on, how they can act and work with their own feelings to limit the impact on their life. Does that all make sense? Yeah. Okay, so a couple of cases. I've chosen a couple of ones that people have seen recently. So the first one I wanted to tell you about was it was when I was on the home treatment team. So I was called out to someone's house, a lovely Eritrean family. They all kind of gathered round, and I was offered a very nice cup of Eritrean coffee, which is very, very strong. I couldn't refuse it. Normally I say no, but they were very, very insistent. The daughter in the family, she was an adult daughter, and she had a child of her own, and usually kind of lived separately. But she had just changed in the last few months. She'd stopped seeing other people, stopped interacting with her friends. She stopped looking after her own child, and she was absolutely terrified that something was happening out there. So she was at kind of a really early stage of becoming psychotic, and it took a lot of work with the family kind of to build up trust. She was really, really suspicious of everyone, especially of me, because I was a stranger and I came in a suit. So but there we had the family on board. One thing that's really kind of crucial to highlight there is how important families are and carers are in forming a kind of a bond with people. I think, as someone pointed out earlier on, it was you too, people don't take medications. We know that it's not just psychiatric medications, blood pressure medications. People don't take them. So a crucial part of what we do is building up trust and engaging with people to help improve kind of compliance and concordance rates with medication. Because we know that helps people, and involving the families, and that's really important. So what I was doing there was, I guess, building up trust, using the family. I did try to engage with her in a kind of a psychological way as well. I gave her an example that I often use of someone standing at a bus stop. If you're feeling sad and kind of a bit anxious and you see someone laughing, you might interpret it as they're laughing at me. Whereas if you're happy, you might see someone laughing and you will think, oh, they're just laughing because they're happy as well. So it's a kind of a way of reasoning. Although it was early stages, it's beginning that kind of psychological approach to thinking about someone's symptoms, and it was also helping the family to think about it in that way and kind of normalising a little bit. So this isn't something really bizarre and alien that's happening. This is an experience that we can all have. And indeed, I think it is a relatively normal experience. I think 1% of the population experience hallucinations, and far less are diagnosed with psychosis and schizophrenia. Second case to talk about is a man I saw. He'd just come over from France. He'd been in Belgium for 16 days. He left France because he felt he was being harassed there. He left Belgium because the European Court of Human Rights couldn't do anything for him. He actually went there to try to insist upon something being done. Wasn't quite sure what. And he was kind of staying with a family member who happened to live in London. Started writing letters to the Prime Minister, to the House of Commons, insisting that people were kind of watching him, and he wanted something to be done about that. There was false perceptions or misrepresentation of him being kind of spread around somewhere. And then he started to become convinced that people were kind of hacking his internet and kind of following him around. So when I saw him, the first time we he was also using a vast amount of cannabis and crystal meth, which may have been contributing. So, and this is another thing, we see a lot of this. It's often very difficult to disentangle kind of drug use and psychosis. And indeed, sometimes they can't be disentangled, the two kind of do go hand in hand. So our job was to try to work out what was going on. And he was again really suspicious of who we were, what we were doing, why we'd been brought there. And the way we kind of approach that, I mean, one is obviously kind of building up trust and trying to explain who you are. But when we're kind of taking a history or speaking to someone like that, we're kind of like a detective in a way. Our job is to kind of, when someone's talking, you kind of notice like little things that they say which are potential symptoms, and you'll kind of pull at the string and tug at it, and you'll get more of that. And we ask kind of questions like, how is that happening? Why is that happening? Who's doing that? So really quite open questions that are targeted at specific things they're saying. And that's how we kind of build up a picture of what's actually going on and finding out what these symptoms that we need to know about to make a diagnosis are when we're dealing with someone really suspicious. What happened with him in the end? So our first lady, she was persuaded to try an antipsychotic in the community. It helped, I think, partially, but it put her in a place where she could engage in kind of a therapeutic way of thinking about her symptoms, so with psychologists, psychotherapists. The second one, so he ended up lighting a fire in the middle of his flat. Fortunately, no one was hurt, but it's incredibly dangerous stuff. So we thought, right, we probably can't manage this person at home anymore. So we did a mental health act assessment, and there's always independent doctors and independent social workers when we do that, and they agreed that the person should be brought into hospital. So they were brought into hospital. We were able to try him for a while without them being on any crystal meth and cannabis. We could see how much that was contributing, and then we were able to give them the trial of an antipsychotic medication. And I think it's always hard when you're sectioning people. It's a really difficult part of the job. And one of the questions that we're always asking, that I think about, is who do I owe the duty of care to? Is it the person now who's telling me I don't want to be sectioned, or is it them, say, a few months ago or years ago when they were well, and what would they think of things as they are now? So it's kind of an ethical quandary, but we do go down that route. And finally, the big challenges, I think, for the profession of psychiatry. We saw stigma against psychiatrists, I think, earlier on. That's fine. That's and I think we've done things to deserve that in the history of psychiatry. But I think stigma to patients, I can go home at the end of the day. With patients, it's with them all the time, and it's not just stigma from other people. It's stigma, it affects how they think about themselves as well. And so I put up there, and that's a. Does anyone know who Frank Bruno was? Yeah. Okay. Good. So he just in case, he's a really famous boxer from probably before some of you born. But that was a headline from The Sun. I had to cut down that image because there was the usual kind of Sun stuff at the top. But but it's the headline though, which is the thing we're supposed to concentrate on. It's awful. It's bonkers Bruno locked up, and there was an outcry. Which I think this is the wonderful thing that there was an outcry, because Frank Bruno was I think the hero too, and they described him in a later article as a hero. And they republished the paper on the same day, I think, after the outcry, with a much more kind of calmer headline, something like I can't remember what it was, but it was more like tragedy as Bruno is admitted to mental hospital, hero battling against illness, or something like that. But this is the kind of stuff that people see daily, and it's still, just because that headline was a long time ago, it's still going on. And I think that also plays out not just to patients who are kind of less likely to seek care, less likely to kind of accept or want to accept that there's an illness going on. It also plays out in terms of how services and policies are kind of playing out. So I've put something out, kind of bed pressures and cuts. They're happening everywhere in medicine. You've seen all the kind of headlines about A&E. Psychiatry, though, is funded far, far less in terms of need than physical healthcare. Almost all the money goes into acute hospitals. The only areas of psychiatry that is easier to get funded in are kind of liaison psychiatry, where they work in acute hospitals and they can relieve some of the pressure on A&E. So, as an example of that, I sometimes have to see people and we don't have any beds, not just in my hospital, but in all of London. So I remember once having to work very hard trying to reconcile a family to the fact that someone might have to be placed under reception. It took a lot of work, and then I went back to the kind of A&E liaison office and spoke to the nurse and said, oh yeah, but there's no beds. We're going to have to admit them to, where was this? It was somewhere up north, somewhere like Whitstable, and I couldn't believe it. And I think there would be an outcry if that happened in, say, cardiology. There were just no beds in all of London for patients. But that happens. So it's affecting our patients more than most. And the third point I wanted to say, I think again it does relate to stigma. The physical health of our patients is a huge issue. Does anyone know how much earlier our patients die than the average member of the population? Yes?
About 20 years.
Yeah, 20 years. And that is shocking. And it's not from suicides, it's not from their mental health problems, it's from physical health conditions. And I think there's lots of things going on there. I think partly it's the fact that people are less likely to access care. There are obviously, aspects of the medication and the associations of the condition, things like the fact that people smoke more in particular, that contribute to that. But I think there is also something about the way our patients are perceived, both from psychiatrists and from medical doctors. So GPs and doctors are less likely to ask people with mental health problems about their physical health conditions. I remember someone in A&E who was being transferred, actually I was trying to transfer them back to A&E. It turned out they'd had a relapse of their schizophrenia because they were in renal failure. And that happens, but they didn't have any blood tests in A&E because it was just assumed that they had psychosis. I think the reverse is true. We as psychiatrists don't do as much as we should do with physical health. For example, we haven't, smoking was only banned in our hospitals, in the hospital I work at this year, whereas in medical hospitals there's been no smoking for a very long time. So we need to do better as well. And I think there are kind of structures in place which reinforce that. Mental health trusts are separate to physical health trusts, and they're the only kind of medical specialty where there is a completely different organisation responsible for someone's care. Everything else happens together. So right from the kind of outset where it's a disadvantage, because we're treating people in silos. And that's what this concept of parity of esteem is all about. It's all about treating mental health conditions with the same level of importance as physical health conditions. So those are my three big challenges for us as a profession. But I still, despite the fact that there are challenges, I love my job. Every patient is unique. Unlike, say, cardiology or it's not just chest pain, it's filtered through that person's lived experience. So every symptom is different, even if it is a hallucination again, it's always unique. It's evidence-based. What we are doing does work, both the kind of medication-based treatments and the psychological treatments, and it's always better together. So it's a research-based field. We work across disciplines. I love that part of it, that it's not just us as the doctors, we're working in several different areas. It's really varied, interesting, rewarding. I hope that's come across. And brings together a whole range. Whatever you're interested in, I think, can be brought into psychiatry, whether it's kind of computational research or at arts and philosophy. There are arts group. There's an arts and psychiatry group at my hospital. There's a philosophy and psychiatry group, both at the Royal College of Psychiatrists and at my hospital. So it brings together a whole range of different things. So I will leave it at that. Do you have any questions?
Thank you. Is there any research working on the relation between thought disorder and interruption and neuroplasticity?
No idea. I'm a clinician, so my research has been in medical education, how we can move groups of doctors from being, good at what they do to better at what they do. I don't work in that field, so I really can't answer your question. I don't know if either of you two could, you might, thoughts?
Thought disorders are the least investigated for symptoms, I think. So I don't like that.
Yeah, so you stumped the audience of experts.
Do you see a huge difference in one person's set of symptoms in a clinical and care setting?
Yeah, we do. And I think this is one of the... We don't do enough of our assessments at home. I think, like all medical specialties, I think a lot of it is hospital-based. Even though psychiatry is better than most that we have, we moved to the community in the 80s, and so medical specialties are trying to do that a lot more now. But yeah, we learn a huge amount. So just as an example, I can see someone, for example, who is, say, manic. They're really elated, quite chaotic in what they're doing, trying to do loads and loads of stuff at once. You get one side of it from seeing them, in your office. When you go to their house, it kind of plays out. Their symptoms play out all over the house. There's things piled up everywhere. The kind of bathtub is full of water and clothes. There's no room to walk. There's no food in the fridge. There's like a bit of food they made from 3 days ago, which they still haven't remembered to put in the fridge. So yeah, you learn a huge amount from that.
Okay. Before I go ahead with the question, I would just like to again reinforce about the Neuro Art exhibition. After having heard your talk, it's just so clear how much is needed still to be done and to support, to remove the stigma and to raise funds for further research. Then next question. Well, the question actually. I was wondering, is there any psychotherapy at the moment that is maybe tried? Because one of the main issues in psychosis is that patients do not have lack the insight of their own condition. To maybe while they are not during the psychosis, so maybe before a relapse, to be educated maybe with the research, with the data that are there now, to maybe try to explain to them what goes on in the psychosis. Would that have
Absolutely. That is exactly what the psychotherapy that we provide our patients is trying to do. It's trying to give people an alternative framework for understanding what they're experiencing. So, and there's different ways of, some people may be very kind of fixed in their beliefs, but that doesn't mean you can't work with them in a psychotherapeutic way. You say, okay, when you have these beliefs, what are the things that make them more likely to come on? So you're kind of identifying, say, triggers, early warning signs, so when they know things are getting worse with them. So you're putting together a framework of their symptoms alongside this kind of alternative approach. And we use, that example of the kind of bus stop thing that I use. You try to relate that with so that's quite an abstract example, but you try to find examples from their life where you're looking at things where they might be kind of misinterpreting things. Generally, when people are well, I think insight is sometimes regarded as an all or nothing thing. People either have it or they don't. But it's actually a kind of a really kind of, there's a big gradation of what insight means. And people do become kind of less certain about their symptoms. Sometimes they will just develop alternative explanations themselves. They'll be like, oh yeah, there definitely were people following me, but now they've stopped because they're following my neighbour instead. Now I just don't worry about it. It's not bothering me. So and then you can work with that. You don't have to persuade them that people weren't following them, but you can work with that approach. So yeah, that's what we're doing. And it's not just kind of psychotherapy. There are loads of patients go to things like hearing voices groups where they will just sit and speak to other people who are hearing voices and talk about their experiences. There's loads of different ways of doing that.
Thank you.
Thank you very much.